Hi, this is a video that I'm starting on. This DNA replication, and our focus would be on leading and lagging strand because sometimes it is very difficult to understand the strands during the replication, that which one is leading, why it is leading, and which one is lagging and why it's lagging. So in order to understand that, let's focus on the DNA structure first. Just review the DNA structure, and then we can go to the next step, which would be the 5 and the 3 prime ends of the DNA strand and what it is, because that also gets confusing sometimes. So in order to understand the DNA structure, let's just review that the DNA replicates uh, in the S phase of the cell cycle, and that's the synthesis phase. So the DNA has four, let's write it here, DNA has four nucleotides. Okay, and this nucleotide is further divided, or it has subunits. Let's put it this way, as three subunits. Okay, so DNA contains a sugar, which is called also pentose sugar, because it has 5 carbons in it. A phosphate group, and one of the 4 nitrogenous bases. So let's put nitrogenous bases. So it's A, T, G, or C. So this 3 makes a nucleotide. And why it is 4? Because we have 4 bases. So we can say we have a nucleotide A as a sugar plus a nitrogenous base, which is A, plus a phosphate group. Okay, so that is 1, and then the sugar plus T, which is another base, and a phosphate. Similarly, we have sugar plus G plus a phosphate, and the last one is sugar plus C plus phosphate. So that makes it 4 nucleotide. Okay, so now let's talk about the structure of the sugar and the nucleotides. So the sugar is a pentose sugar. So I'm trying my best to draw, I'm not good at it. There's a pentagonal shape and there is an oxygen right here. And like we said, it's pentose, so it has 5 carbons. 1, 2, 3, 4, is another carbon. So the structure is like this, 5. Okay, so this is a sugar, and then it has a phosphate group. Let me change the color. So the phosphate group is attached right here, okay, to the carbon number 5 phosphate group. And now we have to connect the base, nitrogen space. So with the carbon number 1, the nitrogen's base, it could be either A, T, G, or C. So let's put A, and then this is connected to another nitrogen space, that would be T, and this is also connected to carbon number 1 of the pentose sugar, but here the structure would be like this. It's inverted pentagon. Okay, so this is C1, C2, C3, C4, C5. Okay, so this is how it makes a strand, and to this C3 there is an OH group, and we'll talk a little bit more about it later in this video. It's called 3 prime, and this is the 5 prime free phosphate, and this is 3 prime free sugar. Okay, so now about the structure, let's magnify this. The adenine, which is a 2-cyclic compound. There are 2 cyclic compounds, so the structure is like this. Okay, there's a double bond here. So adenine and guanine, A or G, they are also called purines, and they have 2, their structure is they're 2 cyclic. Okay, and thymine and cytosine, it has only, the structure is like a hexagon. Okay, so it has, actually I meant to say 2 rings, and it has 1 ring. Okay, so adenine and guanine and purines, and cytosine and thymine, they are pyrimidines. And here there are 5 carbons and 4 nitrogen for adenine and guanine. It's a bigger molecule than thymine and cytosine, Okay, and it has 4 carbon and 2 nitrogen. Okay, there's another difference between this is the adenine, when it bonds with thymine, there's 2 hydrogen bonds, H bonds, and when guanine and cytosine they bond, there are 3 hydrogen bonds. Okay, so this is a little bit stronger than that, so it's easy to break this bond, and we'll talk about it a little bit later when we start the replication process. So we are trying to understand the structure and why it is important to understand the structure, because we are naming 5. You'll see in the textbooks or anywhere about the replication 5 prime 3 prime, and it's important to understand that why it is called 3 prime and 5 prime. So 5 dash, this dash is called prime, so 5 prime, and what it is doing is naming the carbon atom. Okay, and 3 prime is also naming the carbon atom. Why we add prime, why we say prime, not just 3 and 5? The reason is, like we discussed just a little bit earlier, the nitrogenous bases, okay, also contains carbon. Okay. So to distinguish between the carbon here from nitrogen's base, between base and sugar carbon, we name it 5 prime and 3 prime. So when we are talking about replication, you have to think or just visualize in your mind that the carbon we are talking about is of the sugar and not of the base, nitrogenous base. So that's the reason it's called 5 prime and 3 prime. Okay, now we're going to discuss a little bit in this video about the DNA, the generic structure. Normally we see the DNA molecule like that, double helix. Okay, and we'll discuss in more details about the replication. But right now we are just going to just talk a little bit in this video because of the time constraint, that which one is 5 prime and which one is 3 prime. Okay, so this is a double strand structure, and You have to name it 5 prime, this is 3 prime, and this is 3 prime and 5 prime. So these two strands, they are running in opposite direction. Okay, so that's why they call anti-parallel. And these two strands are connected each other by those hydrogen bonds right here we just talked about. So now this end is called a free phosphate end, and this end is called the carbon 3 sugar free end. So why we call it that way? If you look at the bonding here and see this structure I have drawn right here is the 5. So you just imagine that this is the top of the DNA molecule. So this phosphate is not connected. This phosphate group is not connected upwards to anything. Okay, it's only connected to the carbon number 5 and not anything up. And this carbon number 3 is connected to a phosphate group, and this phosphate group is again connected to C5 of the pentose sugar. Okay, like it's up there. So this phosphate group is connected up and also down. Okay, so that you have to understand. So this is C1, C2, C3, C4, C5. So this phosphate group over here is connected, so it's not free, it's connected, because this is, see, this the strand is a continuous strand going down. Okay, so again the same way C3 is again connected to a phosphate group, and then that phosphate group is connected to carbon number 5. That's how it's going down, and it's going this way horizontally between the bases. So it's AT, and it could be GC, TA, because A always bonds with T and G always with C in DNA. So now let's talk about the 3 prime end. Okay, so here if you see, this is the 3 prime end. When this carbon 3 molecule is not connected to a phosphate group like it is connected over here, this end is free. It has only a hydroxyl group, OH. So whatever you do, if you keep stacking more and more nucleotide on top or at the bottom, you'll always find there is one phosphate group free and one carbon number 3, that group is free and it's attached to hydroxyl group. Okay, it's never ending, so and that's how, if you try to imagine the structure, that's how it is. So this is 5 prime and this is 3 prime, and that's what I have drawn it over here, and this is the 5 prime that I have drawn over here. It's free end. Okay, so similarly, the when you look at the bottom, is the same way. So when it's in the bottom, just imagine that this C3 carbon 3, so this is the end, and there's nothing here. There's only a hydroxyl group like we showed in the top. So that's how it is in the bottom. Okay, and same way as the 5 prime end. So that's basically the I wanted to cover in this first video of uh, the leading and lagging strand. So now in the next video, when I talk about the 5 prime and 3 prime end and how the DNA polymerase is adding those nucleotides, so which way it is going, and how it is read, which direction, 3 prime to 5 prime end, and actually it's adding 5 prime to 3 prime end, because it gets a little confusing, and Okazaki those segments how it is adding. So that's why it is very important to understand the 5 prime and 3 prime and why it is called. Okay, so I'll see you in the next video, which would be labeled as DNA replication, leading and lagging strand part 2. Okay, thank you for watching.